Welcome back to Clinical Hematology Reviews. Today we'll be discussing complete blood count, or CBC, principles. Automated CBC instrumentation represents the core analytical engine of the modern hematology laboratory, integrating electrical impedance, optical light scatter, radiofrequency conductivity, and spectrophotometry.
Understanding these foundational principles is essential for evaluating cell counts, interpreting histograms and scatter plots, resolving instrument error flags, and ensuring accurate patient results.
Laboratory testing is divided into three distinct operational phases: pre-analytical, analytical, and post-analytical. The pre-analytical phase begins with proper patient identification, collection of whole blood into evacuated tubes containing dipotassium or tripotassium EDTA, and verifying specimen integrity by checking for microclots.
Once the sample enters the analytical phase, the automated analyzer performs direct cellular counting, hydrodynamic focusing, chemical lysing, and spectrophotometric absorbance measurements alongside internal quality control checks. Finally, the post-analytical phase focuses on data verification, applying the rule of threes, evaluating histogram morphology, and releasing validated clinical reports into the electronic medical record.
Inside the analyzer, the automated fluidic pathway begins by aspiring a microvolume of approximately 15 microliters of whole blood. This aspirated blood is immediately mixed with an isotonic, electrically conductive diluent before being partitioned into two separate analytical streams.
One stream is directed into the WBC and hemoglobin bath, where a lytic reagent is introduced, while the second stream enters the RBC and platelet bath, containing only conductive diluent. This architectural split allows simultaneous, independent analysis of white blood cells and hemoglobin in one chamber and native red blood cells and platelets in the other.
Two foundational reagent systems drive automated hematology cell counting analyzers: the conductive isotonic diluent and the lytic reagent. The diluent matches physiologic osmolarity and pH, preventing red cell swelling or crenation, while providing the high electrolytic ion content required to conduct electrical current across a sensing aperture.
In contrast, the lytic reagent contains quaternary ammonium salts, or non-toxic surfactants, that rapidly destroy non-nucleated red blood cell membranes, reducing them to microscopic stroma. Simultaneously, the lyse collapses leukocyte cytoplasm around intact nuclear membranes for volumetric sizing and converts liberated heme into a stable chromophore for optical measurement.
The primary counting mechanism across modern hematology systems is the Coulter principle of electrical impedance. Blood cells suspended in the conductive electrolyte solution are drawn through a precision microaperture situated between an internal and external electrode, powered by a constant direct current or DC power supply.
Because blood cells are poor electrical conductors, each cell that passes through the aperture displaces an equivalent volume of conductive electrolyte, creating a momentary spike in electrical resistance, where the pulse height, or amplitude, is directly proportional to cell volume, and the frequency of pulses per unit time indicates cell concentration.
Standard aperture flow can introduce physical counting artifacts, such as cells passing along the aperture perimeter or recirculating back into the sensing zone. Hydrodynamic focusing solves this problem by surrounding the central sample stream with a sheath reagent flowing under strict laminar conditions.
The dynamic pressure acceleration narrows the sample core into a single cell diameter column, forcing erythrocytes, leukocytes, and platelets to pass through the sensing zone in strict single file. This eliminates aperture border zone errors, prevents recirculating eddy currents, and avoids coincident passage where two cells might otherwise be counted as a single large pulse.
In the dedicated WBC and hemoglobin chamber, the lytic reagent strips erythrocyte membranes and shrinks leukocyte cytoplasm, allowing white blood cells to be counted and sized via direct current impedance pulses. Particles registering above approximately 30 femtoliters are enumerated as white blood cells.
In the very same chamber, the liberated hemoglobin undergoes a chemical reaction forming a stable chromophore measured spectrophotometrically at 525 nanometers. The optical absorbance directly correlates to total hemoglobin concentration.
In the RBC and platelet chamber, intact blood cells suspended in isotonic diluent are counted without lytic interference. Erythrocytes are sized between approximately 25 and 250 femtoliters.
Platelets are simultaneously sized in the lower pulse range of 2 to 30 femtoliters, with raw channel counts fitted to a mathematical log normal curve to produce a final platelet count. This mathematical curve fitting successfully excludes low-end electrical noise and high-end microcytic red blood cell fragments, while allowing derivation of the mean platelet volume.
Point-of-care or POC systems utilize post-lytic electronic volume sizing to generate a 3-part leukocyte differential histogram. Because the lytic reagent collapses cellular cytoplasm, the resulting impedance pulses reflect nuclear volume and membrane resistance rather than native cellular dimensions.
The histogram divides cells into 3 discrete sizing regions. The lower discriminator to trough 1, spanning 35 to 90 femtoliters, isolates lymphocytes. Trough 1 to trough 2, from 90 to 160 femtoliters, captures the mixed population of monocytes, eosinophils, and basophils, and trough 2 to the upper discriminator, spanning 160 to 450 femtoliters, measures neutrophils.
High-throughput laboratory analyzers provide a comprehensive 5-part differential using VCS technology, analyzing leukocytes in their near-native states without harsh cytoplasmic stripping. VCS stands for volume, conductivity, and scatter, combining 3 distinct physical probes into a single measurement flow cell.
Volume is measured using low-frequency direct current to establish accurate physical cell displacement. Conductivity applies high-frequency radio current that penetrates the cell membrane to probe internal nuclear size, lobularity, and cytoplasmic chemical density, while multi-angle laser light scatter measures external surface structure, granularity, and internal refractivity.
When raw VCS data is plotted on a multi-dimensional scatter plot of volume versus side scatter and volume versus opacity, distinct leukocyte populations separate into well-defined topographical clusters. Lymphocytes cluster in the lower left region due to their small volume and minimal internal granularity.
Monocytes appear above lymphocytes with higher volume and moderate scatter, while neutrophils occupy the mid to upper central zone with moderate volume and high scatter from their secondary granules and segmented nuclei. Eosinophils separate toward the far right due to their highly refractive, intensely light-scattering granules. Basophils resolve on the conductivity plane between lymphocytes and monocytes, and nucleated red blood cells fall below lymphocytes near the noise threshold.
Hematology analyzers incorporate sophisticated error codes to alert the laboratory scientist to analytical or mathematical abnormalities. A display of plus signs indicates that a parameter exceeds the operating linearity range, requiring a manual 1:2 dilution with isotonic saline, a repeat run, and multiplication by the dilution factor.
A series of dashes indicates a total vote-out, where sensing apertures disagreed statistically, necessitating fluidic inspection and a sample rerun. An R flag signifies a review result where a parameter was derived from unstable or flagged raw data, blocking automated reporting until the laboratory scientist inspects raw channel histograms or reviews a stained peripheral blood smear. Colon marks indicate a flow cell clog requiring an immediate backflush. Question marks reflect display buffer overruns, and dots indicate incomplete computation caused by an upstream primary failure.
Quality assurance requires a structured maintenance schedule divided into daily, periodic, and lot validation tasks. Daily and per-shift procedures mandate startup fluidic flushes, electronic aperture background counts to ensure reagent purity, and analyzing 2 to 3 levels of commercial quality control material within 2 standard deviations.
Periodic protocols include weekly and monthly deep cleanings with chemical deproteinization of baths and line decontamination to prevent protein accumulation. Lot validation involves daily reagent verification alongside rigorous lot-to-lot validation studies prior to placing new diluent or lysing reagent lots into clinical operation.
The step-by-step analytical procedure begins by confirming acceptable startup background limits, such as a WBC count under 0.2 × 10 to the third per microliter and a background platelet count under 10 × 10 to the third per microliter, and verifying that quality control runs are in range. Next, the pre-analytical specimen inspection requires inverting the EDTA tube 8 to 10 times and gently sweeping 2 wooden applicator sticks through the sample to confirm the absence of fibrin clots.
After presenting the specimen for automated aspiration, the analyzer performs internal dilution, dual-chamber partitioning, impedance counting, and 525 nanometer spectrophotometry. The laboratory scientist reviews the numerical data and scatter plots, applies the rule of threes, where red blood cell count times 3 approximates hemoglobin, and hemoglobin times 3 approximates hematocrit, executes any necessary corrective actions, and releases validated clinical results.
Biosafety in automated hematology mandates strict adherence to standard precautions, treating all blood specimens as potentially infectious for blood-borne pathogens, including hepatitis B, hepatitis C, and HIV. Personal protective equipment, including nitrile gloves, fluid-resistant laboratory coats, and safety glasses, must be worn during specimen handling, uncapping, and analyzer maintenance.
Closed tube sampling should be utilized whenever possible to eliminate aerosol generation during piercing and aspiration. Furthermore, all contaminated wooden applicators, transfer pipettes, and gauze must be disposed of immediately in rigid biohazard containers, while chemical effluent containing quaternary ammonium compounds must be managed in accordance with hazardous waste regulations.
Today we covered the core principles of automated CBC analysis, spanning dual-chamber fluidics, electrical impedance, VCS leukocyte differentiation, mathematical indices, instrument flagging protocols, quality control, and laboratory biosafety.
Thank you for joining us, and we look forward to having you with us next time.